Transmission of genes from generation to generation. This should be a review. You guys should have learned how to do Punnett squares and stuff in regular bio. You guys are actually going to do a discussion this week, excuse me, on genetic screening. You're going to watch a video about a woman named Patricia Stalling. She was convicted of killing her infant son. And you're going to answer questions about possible implications about her situation. Genetic screening actually got her conviction overturned. So we'll learn a little bit about that. But how are traits inherited? Before Mendel, there was no clear understanding of how traits were inherited and passed from one generation to the next. Working with pea plants, Mendel used experimental genetics to uncover the fundamental principles of inheritance. So you guys did a little bit of background on Gregor Mendel a couple weeks ago, or maybe it was last week. I don't remember which week it was. But you guys learned about Mendel. He was a monk. He was from Austria. He worked with pea plants. He was a little bit OCD to have to work with pea plants and count thousands of numbers of flowers and pea plants and keep track of their colors and their shapes. He was a little crazy, but he is the father of genetics. Mendel's success in discovering the fundamental principles of inheritance was the result of carefully planned experiments, and his first step was selecting an organism. He defined ideal properties for experimental organisms used in genetic studies today. So when geneticists study something new, they want to study a new principle or a new trait, they still go back to Mendel and say, okay, why did he pick the organism that he picked? Advantages of pea plants. So this is why he chose the plants that he did. So there's many different varieties available, each displaying a distinguishing trait. It was all either or. For the pea plants, for the actual peas themselves, it was either green or yellow. The flowers were either white or purple. There was nothing in between. It was really easy to keep track of traits that way. The plants were capable of self-fertilization or artificial fertilization. He could fertilize them himself. They had flowers that reduced accidental pollination. The offspring were fully fertile, allowing more crosses to be performed. Pea plants had a short life cycle and could be grown outdoors or indoors in greenhouses when it got too cold, because where he lived, it snowed. He would not have been able to continue his experiments because it was too cold and flowers wouldn't grow. So he had to build a greenhouse and grow them himself. He could do this all year round. Mendel studied how 7 different traits were passed from generation to the next. His initial crosses studied the inheritance of a single trait, such as seed shape or seed color. So his 7 traits, the shape, color, pea coat color. He studied the pods, the shape and color of the pods that the peas came in. He studied the flowers, the position of them. It could either be axial along the stems or terminal on the top of the stems. And the stems could either be tall or short. So we usually just look at either the flower color or the pea shape and color, but he actually studied 7 different traits with his OCD-ness. So for his first experiment, plants with smooth peas were crossed with plants with wrinkled peas. So you can see in the picture at the top, a smooth was crossed with a wrinkled. That first cross is called the pea or parental generation. You'll see these terms a lot as we go through genetics. All the offspring, he noticed, produced smooth peas. None of them were wrinkled. And that first generation is called F1, generation 1. Then he took a bunch of the smooth peas, the F1 generation, and crossed them with each other And those are the numbers he got. He counted all thousands of those smooth versus wrinkled peas. And what he ended up with was a ratio, a 3-to-1 ratio of smooth to wrinkled peas. So for every 4 peas, 3 of them were smooth and 1 of them was wrinkled. All of Mendel's monohybrid crosses yielded the same results. The F1 generation displayed only 1 parental trait, and the trait not present in the first generation showed up in the second generation at a rate of 25%. 1 out of 4 is 25%, just like quarters. And this result was seen regardless of which trait was contributed by the pollen. So for seed shape, for seed color, coat color, pod shape, pod color, all of these numbers stayed roughly the same. He always got that 3-to-1 ratio when he crossed that first generation of offspring. Genetic factors, now known as genes, can be present but not expressed. This is one of his main conclusions. The expressed trait, which was that smooth P, is called dominant, and we use capital letters to show this when we write them down. The unexpressed trait, which is hidden in that first generation, the wrinkled, we use lowercase letters to show that it's recessive. So Mendel concluded that despite the identical appearance of the parents in that first generation, they were both smooth. Because of that second generation, he figured out that the parental plants in the first generation must be genetically different. Something about them inside must be different, even though they look the same. And although pea plants have 2 genes for each trait, they contribute only 1 to their offspring because of meiosis. Mendel didn't know that at the time, but we now know that our bodies and plants do this and animals do this. They cut our gametes in half, our sex cells, so we only contribute half of the information to our offspring, so we don't double it every generation. A couple more conclusions. Each plant carries 2 genetic factors for each trait, 1 from each parent. And in the pea plants, traits were not blended as they passed through the parents. It was either or. It was either wrinkled or smooth. There was nothing in between. All right, some genetic definitions, phenotypes and genotypes. Phenotype is the observable properties of an organism. It's what you can see on the outside. It's a thing's physical characteristics. So phenotype, think they're physical characteristics, what you can see on the outside. The genotype is the specific genetic constitution of an organism or what is found on the inside of their cells. So if you look at the picture, the phenotype would be either smooth or wrinkled. It's what you can see on the outside of the pea. And then the genotype are these little letters that we assign that are either dominant or recessive. We're going to be doing a lot of this. So You'll get a lot of practice. An allele is an alternative form of a gene, usually distinguished by its phenotype, or its phenotypic effects, like brown eyes versus blue eyes in humans, or smooth versus wrinkled in peas. It's the same exact gene, but it's a different version of it. And that is pronounced allele. Genotypes can be homozygous, which means they have the same alleles for one or more genes. So they would either both be capital or both be lowercase. The prefix homo means same, so that should be easy to keep track of. A genotype can also be heterozygous, meaning they have two different alleles for one or more genes. And if you'll notice, they're both A's. For the example, we use different letters. But they're two different versions of that letter. So you're always using the same letter for genotypes, to write out genotypes. They just might be different versions. And those different versions are called alleles. Mendel's first law. So he actually got to make laws. He got past theory and got to make laws. Individuals carry two genes for each trait, but pass only one gene onto offspring. How is this possible? What's that process called? In the picture, where you have one and it ends up in four, and you cut the information in half. The members of a gene pair separate during gamete formation, which is the process of meiosis. It's the gametes of adults, their eggs and sperm, that go on to make offspring of the next generation. And all of this is called the principle of segregation, because those alleles, those chromosomes, they segregate randomly into those 4 cells you see at the end. So a diagram called a Punnett square can be used to visualize and keep track of all possible combinations of alleles, phenotypes, and genotypes in genetic crosses. And there are steps to putting together a Punnett square. I broke down all the steps, or I put them into steps. Once you get used to them, you don't have to think about the steps quite so much. But if you don't remember how to do them, always come back and follow the steps. So step 1, you got to determine the genotype of the parents. And there are always 3 options. They can either both be dominant, which we call homozygous dominant. The phenotype is also dominant. And for our example, we're going to use the smooth peas. The genotype could also both be recessive. The title for that is homozygous recessive. We use lowercase letters, like I said. And the phenotype would turn out recessive, the physical characteristics. Physically, it would turn out wrinkled, because wrinkled is the recessive trait. Or a genotype could have one of each allele, where you use one of each letter, a capital and a lowercase. We call that heterozygous because they are different. But the phenotype is dominant. That dominant or strong allele will mask the weak one. And this is a very important slide going forward and trying to determine Punnett squares. So you might want to star it or highlight it or bookmark it somewhere. So for our example, both parents are heterozygous and they hold one of each allele in their genes. They both look smooth because when they're heterozygous, the dominant condition is always shown. But their letters on the inside, their genotype, is one of each. You have a big S and you have a small s. Both parents here are the same. So now what process do the parents need to go through to cut their genetic information in half? We've said it like 3 times. You need to go through meiosis. So step 2 is meiosis. Gamete formation is used to cut genetic information in half. So in our regular cells, they're put together. In our gametes, everything is halved. So step 3 is you set up the Punnett square using the gametes from meiosis. You put one parent on each side. Doesn't matter which parent goes where. Here they have the female on the side and the male on top. You put one gamete next to each box. And this is called a monohybrid cross because we're only crossing one trait. We're just talking about the smooth versus wrinkled. Mono, the prefix mono means one. We're only looking at one trait. And then step 4, you bring the top letters down and the left letters across to mix the parents' gametes. I wish we were in class because it's a lot easier for me to show you on the board, but that doesn't work right now. So I put a GIF there. This is how it's done. You can see the big B goes down and the small b, these b's go across. So these go down, these ones go across. Then you should be able to analyze the phenotypes and the statistics of the children. The 4 boxes in the middle represent possibilities of children's gametes. So we paired this S and this S together. So this first box has 2 capital S's. We pulled this big S over here and paired it with this small s. We pulled this big S down here, paired it with this small s, mixed them, and then this one and this one are both small. So here's the overview that we did. We had the smooth versus smooth. These are the parents. There's their gametes. We set up the Punnett square, and then we combined them all. And now we get to analyze them. Oops, sorry. So 3/4 of these children, or I'm sorry, the children have 3/4 of a chance, or 75% of a chance, of coming out smooth. Those are the phenotype ratios. They have 1/4 of a chance, or 25%, of turning out wrinkled. You could also analyze the genotypes. The children have a 25% chance, a 1 out of 4 chance, of being homozygous dominant, where both of their alleles are capital. They have a 50% chance of turning out heterozygous, where they have 1 of each allele in their genotype. And they have a 25% chance of turning out recessive, where they have 2 small alleles. Mendel also did dihybrid crosses, big ones. Di equals 2, mono means 1, di equals 2. So Mendel analyzed 2 traits with his next cross and made 1 big Punnett square. You guys are actually going to be doing some of these in one of the labs this week. And there are websites to help you get through it step by step. But he looked at both color, the yellow versus green, and he looked at the wrinkled versus round or smooth. So in Mendel's dihybrid cross, plants in the F2 generation showed 4 phenotypic patterns. They could have 4 different phenotypes. 2 of them look like the parents. So there were smooth yellow ones and there were wrinkled green ones. But then you had 2 new phenotypes. You had smooth green ones and you had wrinkled yellow ones. Those were new. And these 4 phenotypes always occurred in a 9:3 to 3:1 ratio. There were 9 smooth and yellow, because those are all of the dominant conditions. On the other side, there was only 1 wrinkled and green. Those are all of the recessive conditions. And then there were 3 of each that were mixed. 3 smooth and green, and 3 wrinkled and yellow. So that ratio is a very famous ratio. 9:3 to 3:1. And the total is 16. So Mendel got a second law. Not only did he get a first, he got a second. The principle of independent assortment. Members of one gene pair separate or assort independently of other gene pairs. So they segregate randomly, and then they assort independently. They don't depend on anything else. Alleles are randomly distributed into gametes during meiosis. In a cross between 2 individuals, there is equal probability for any combination of gametes. And this explains the inheritance of 2 traits. Just because a pea turned out green doesn't necessarily mean it's also going to be wrinkled. Those 2 traits are not connected. They sorted independently during meiosis. Meiosis explains Mendel's results. Gene pairs or alleles are located on chromosome pairs, and the position is occupied by a gene. The position occupied by a gene on a chromosome is referred to as its locus. The behavior of chromosomes during meiosis causes segregation and independent assortment of alleles. So you can see there's 2 chromosomes there. You have 2 different alleles for flowers. You have an allele for purple and an allele for white flowers. But they are on the same location on the chromosome. They're just different versions. The location is called the locus. I don't know why scientists can't just say location, but they shortened it up to locus. Genes versus chromosomes. When Mendel was working with pea plants in the 1800s, the behavior of chromosomes from mitosis and meiosis was unknown. That didn't get figured out until after 1900. In 1903, two scientists independently proposed that because genes and chromosomes behave in similar ways, genes must be located on chromosomes. That is the chromosome theory of inheritance. And there has since been tons and tons and tons of experiments generating data that support that theory. That's why we call it a theory. So each gene is located at a specific site called a locus, like we said, on a chromosome. Each chromosome carries many genes. And it's estimated that 20,000 genes are carried on the 24 different chromosomes that can be had by people. We all have 22 autosomes. There's also an X and a Y. Girls have two X's, boys have an X and a Y. And then there's a table down there, it's also in your book, summarizing the relationship between genes, chromosomes, and meiosis. And there's a picture form of genes separating and assorting during meiosis. Meiosis 1, there's meiosis 2. It has your genotypes in there associated with each chromosome. You can see you start with a parent. You got one homozygous dominant parent and one homozygous recessive parent. With two different traits, whatever trait A and B is. But they go through meiosis and they all end up randomly. They can end up all capitals and all lowercase, or they can end up all mixed up. We have capital lowercase, and then they switch. Mendelian inheritance in humans. We do have some Mendelian traits. Segregation and independent assortment occur with human traits. Albinism is one example that we can use to illustrate it. Albinos carry two copies of a mutant recessive allele. Albinism is a disorder where your body can't make pigment. You can't make melanin. Melanin is the pigment that makes us dark. Some people have a little bit of pigment, some people have a lot of pigment, but everybody usually has some melanin. And albinos, or people with albinism, they don't have that melanin. So we do the same thing, follow the Punnett square. You have two normal parents, but the normal parents happen to be heterozygous. They each have a small a, which is the recessive allele that will cause albinism, but since they have that dominant allele masks the recessive, and the parents over here on the left are normal. They appear normal. And they go through meiosis. Their gametes separate. We do a Punnett square for them. And you can see by the bottom right square that their children have a 1 in 4 chance of expressing the albino disorder, of having albinism. So important, this does not mean that there will be one albino child and 3 normally pigmented children in every family with 4 children. It just means that if the parents are heterozygous, each child has a 25% chance of being an albino and a 75% chance of having normal pigmentation. So it doesn't mean if they have 4 kids, one is definitely going to be an albino. It just means with each child that they have, each child has a 25% chance of inheriting both recessive alleles and having albinism. The inheritance of 2 traits in humans also follows the Mendelian principle of independent assortment. To illustrate, let's examine a family in which the parents are each heterozygous for albinism, and they're also heterozygous for hereditary deafness. Being deaf means you can't hear, and that is also a recessive trait. So if both parents are heterozygous for both traits, we have the one big, huge Punnett square. You double it up with the 16 squares. And the ratio turns out the same as the p's. You have 9 normal children who are normally pigmented and they hear just fine. They are not deaf. You have 3 that have normal pigment but are deaf. You have 3 that are albino, but they can hear just fine. And then you have 1, a 1/16th chance that the child will have both albinism and be deaf. So you're going to do an assignment where you're going to be working through some dihybrid crosses. A pedigree is a diagram illustrating the members and ancestral relationships in a family, using standard form and symbols to show the inheritance of a trait and to estimate the risk of any given family member of inheriting that trait. So the numbers over here are the generations, and the circles and the squares and the fact that they're filled in or not filled in, they all mean something. On the next slide, you have a chart of all of the meanings. So squares are males, circles are females. You have who mates, you have mating between cousins, which used to happen a lot, doesn't happen so much anymore, but it used to happen a lot in the old days. Parents and children, the Roman numerals, the 1 and the 2 symbolize generations, and then the 1, 2, 3 symbolize birth order. So number 1 is the oldest child, number 2 is the middle, number 3 is the youngest. You got twins, offspring of unknown sex, aborted or stillborn offspring, deceased has a line through it. And then the other important part is if there's circles and squares and they're not filled in, they're unaffected. They do not have whatever trait we're talking about. Usually we talk about diseases when you talk about pedigrees. So if they're not filled in, they don't have the disease you're talking about. If they are filled in, they are affected. They have the disease. And then if they're half filled in, they're heterozygous, like the albinism. They have 1 dominant allele that masks the 1 recessive allele. So like I said, each generation is identified by a Roman numeral. If you don't know your Roman numerals, I included the link there. And then each individual within a generation is identified by a number. The offspring are connected by a horizontal line and listed in birth order from left to right along that line. And then a proband is marked with a P. That's the first affected family member who seeks medical attention for whatever trait or disease they're talking about. You guys are going to be analyzing pedigrees. In your quizzes and tests this week. So that chart with the key, with the circles and squares and filled in and blanks is going to be very important for you this week. Following a trait through a pedigree. You guys are going to have to know how to analyze this. Which ones are girls? Which ones are boys? Which ones are deceased? Who has the disorder? So if you go back to this chart, that should help you read through that pedigree. Non-Mendelian genetic traits. So not all traits follow Mendel's principles. So Mendel's traits are all either or, either smooth or wrinkled, either purple or white, either yellow or green. There's no blending. However, there are some traits, especially with humans, that do get blended. We call those non-Mendelian traits. So alleles can interact ways other than totally dominant or recessive. Your incomplete dominance has a distinctive phenotype in the heterozygotes. That's where the blending comes in. There's something called codominance, where both alleles are fully expressed in heterozygotes. And then multiple alleles, many genes have more than 2 allele options. Eye color and hair color are 2 of them. That's why there's so many varieties. And then genes can interact to produce unique phenotypes in a process called epistasis. So incomplete dominance, the expression of a phenotype in a heterozygous individuals, is a blend of the parents. This is not what Mendel studied, but it does happen. So snapdragons is what these flowers are called. If you mix a red one and a white one, you get a pink one. In humans, hair texture is incomplete. If a person with curly hair has a baby with a person with straight hair, there's a chance that the baby will have wavy hair, kind of an in-between texture. Now, some offspring will still be red and white, but if the offspring have a heterozygous genotype, that's when that will be blended. If they have a dominant genotype, they'll still be red. If they have a recessive genotype, it'll still be white. Multiple alleles, blood types. Our blood type is controlled by multiple alleles. The inheritance of ABO blood types in humans is controlled by the dominant A and B alleles. And the recessives use a small I allele. If a person, there's 4 different kinds. If they have both dominant A's or even one dominant A and a small I, they're going to be type A. Same for the second one. If they have all B's or one B and one recessive, they're going to be type B. But if a person has an A and a B allele, both of those are going to be expressed. And we just say they have type AB blood. They're not mixed like incomplete dominants. They're codominant. They both show up. And then if you have two recessive alleles, you have type O blood. I have type O blood. Type O blood is very common. Type AB is the most rare. And then codominance in human blood type, again, blood type is the same. It's codominance and it's controlled by multiple alleles. Cells expressing the IA blood allele, of blood type A, IB is B. Cells having both, we just call them AB. And neither are type O. And these last two slides are going to be important for you in the lab you're going to do this week. So make sure as you're going through the assignments this week, you have the notes with you. Epistasis is a situation where the expression of one gene is affected by the expression of another independently inherited gene or genes. For example, if the expression of gene number 2 depends on the expression of gene number 1, but gene number 1 becomes inactive or doesn't get expressed, then gene number 2 doesn't get expressed either. And we say that gene number 1 is epistatic to gene number 2 because it basically just turned it off. And I think that is it for the notes. So like I said in the beginning of class, you guys have one more chance to do the exam yesterday because so many people had problems with the pictures. But get it done today. It closes tonight. And then move on. You do have the Mendelian inheritance lab with all the Punnett squares. I'm going to post that a little later today. I had to come to school to get some stuff to scan and post for you. So that'll be posted in a little while. But today, if you want to retake the test, concentrate on that today. And then you can work on the rest of the assignments the rest of the week. All right? Questions?
No, I don't have any questions.
Everybody good? Everybody getting the flow of the class? We have notes, you have work, you have a test, and then we move on to the next week.
I would like to say that I'm type B blood because both my parents are both type B.
Well, there you go. Yep, I'm type O, so both my parents were type O. All right, well, that's all I have for you guys. Go forth and start your weekly work. Retake the test if you want to. And I'll see you guys Wednesday. Wednesday's our Q&A session, so come with questions. Question, Maria?
No, have a good day.
Okay, have a good day.
Have a good day.
Which test?
We'll see you Wednesday.
Wait, which test?
What?
Which test? Not the exam, right? You're not talking about that one?
The chapter 2 exam that was due yesterday?
Wait, which one are you saying we can retake? The first one, right?
No,
All right.
chapter 2. So check your Canvas announcements. I made an announcement about it on Canvas.
Okay.
Ari, I think you're in the wrong class.